Now move to the last talk of the day, that is by Peter Besmer. The abstract on page number 20 and 21, and the introduction is actually for the people of my generation, maybe after me. He actually joined Gobin at Madison in 1970 and continued to work with him until 72 at MIT. He then continued to work at MIT in Baltimore's lab until 79, and then came to Memorial Sloan Kettering Cancer Center, where he continues even now. The title of this talk is Oncogenic KIT Receptor Signaling and Targeted Molecular Therapies in Mouse Models.
Of course, I would like to thank Gobin as well as Tom and Asim. Asim I actually knew when he was at Sloan Kettering a little bit, for inviting me, and I came in, as was mentioned, I came to Gobin's lab in 1970 in the middle of January. It was really freezing cold, and but I had a wonderful time here, especially after the iceberg on the lake in the first week of April. And so this is just a view from the Union onto the lake. Now, this is the group. You have seen this picture before, I think yesterday. At the time I was in Madison. There is only one person missing that I know, and that is Ian Molyneux, who was on a trip with his fiancée. And of course, there is Marv in the back there, and then Bob Miller, with whom I worked a lot, and Hans van der Sande, and Kjell, and Bruce, and everybody else. So, of course, at that time, I was also part of the chain gang, as mentioned this morning. Now, as Marv mentioned yesterday, that I was assigned to a wonderful place to live with wonderful friends, namely the house which was built by a pupil of Frank Lloyd Wright, and these are pictures of that. Now, for quite some time, work in my lab was concerned with the elucidation of mechanisms of how receptor tyrosine kinases produce their cellular responses in different cell types in vivo during embryonic development, as well as in the postnatal animal. And as a model, we have been using the KIT receptor tyrosine kinase and its ligand, KIT ligand. Now, KIT is a class 2 receptor tyrosine kinase, which characteristically is made up of an extracellular domain, which is made up of 5 immunoglobulin-like domains and a single transmembrane domain, and the cytoplasmic domain, which includes tyrosine kinase, which is divided up into 2 parts by a kinase insert segment. Now, KIT ligand is a membrane growth factor which may be proteolytically cleaved process to produce soluble KIT ligand. Thus, KIT ligand may actually function both as a soluble factor and as a membrane growth factor, which delivers its signal by cell-cell contact in a juxtaclean fashion. Now, critical steps in our understanding of the KIT receptor system were the demonstration of identity of KIT and KIT ligand with the murine, white spotting, and steel loci respectively. Mutations at the KIT and KIT ligand loci generate deficiencies in several major cell systems during embryogenesis and in the adult animal, namely hematopoiesis, organogenesis, germ cells, germ cell development, and in the gastrointestinal tract. in the development of interstitial cells of Cajal. This is a cell system that we are going to be concerned with. In the postnatal gastrointestinal tract, ICCs, or interstitial cells of Cajal, localized to the myenteric plexus region and to the muscle layers. This is a cross-section through the gut wall, the mucosa, submucosa, and then the muscle layer with a longitudinal muscle layer and a circular muscle layer, and in between is the myenteric plexus. ICC functions as pacemakers in the gut and are mediators of the propagation of electrical slow waves. Mice with loss of function mutations in KIT and KIT ligand lack ICCs, and they may have impaired peristalsis and may have a colon. Now, based on mutant phenotypes and distinctive cell compartments, in vivo, the cellular responses mediated by KIT are quite diverse and include cell proliferation, survival, adhesion, migration, differentiation, radioresistance, and then also neoplasias. Now, originally we identified KIT as an oncogene in an acute transforming feline retrovirus. Today, KIT involvement is known in several human malignancies, including gastrointestinal stromal tumor, mastocytosis, a small subset of acute myelogenous leukemias, germ cell tumors, actually a subset of seminomas, and in a subset of melanomas, acrals and renal melanomas. In all these situations, the KIT receptor has acquired activating mutations. Now, gastrointestinal stromal tumor is the most common mesenchymal neoplasm of the human GI tract. These express the KIT receptor, and they are thought to derive from an ICC progenitor or ICC through somatic mutation of the KIT receptor. A small subset of these have mutations in the related PDGFR receptor tyrosine kinase. Now, this is a schematic representation of the type of mutation one finds in different tumors. The majority of the mutations in GIST are discovered in the juxtamembrane domain. Then, in a smaller subset, mutations in the extracellular domain are found as well, in GIST as well as in AML. And interestingly, these mutations localize to an interface in between the two dimerized receptor chains, and thus it is quite possible that actually the mutations stabilize dimer formation and thus increase receptor activation. In addition, in the N-terminal kinase domain, there are some mutations, and then in the activation loop of the KIT kinase, mutations which are predominantly found in hematopoietic malignancies and seminoma. Now, this is just a summary of the type of deletions and point mutations and simple insertions that are found in KIT exon 11, which is the juxtamembrane domain, in 120 cases studied at Memorial Sloan Kettering by a colleague of mine, Christina Antonescu, and you can see that there are deletions of various sizes, substitution mutations, and insertions. Now, how does this come about? The juxtamembrane domain of the KIT receptor tyrosine kinase is actually an autoinhibitory domain of the KIT kinase, and this is here indicated in purple, and it is quite likely that mutations in this region disrupt the conformational integrity of the autoinhibitory function and thus activate the receptor. Then the activation loop is shown here, which sort of dramatically changes conformation upon phosphorylation of a tyrosine residue in that location. Now, the reason why we actually got very much interested in this was the observation by a Japanese group of human familial GIST syndrome with a germinal mutation in the KIT gene. And this told us that actually you may be able to generate a mouse model because mutation may actually survive embryonic development, or the mouse may survive embryonic development and achieve adulthood. Now, an important consideration here was really that the juxtamembrane domain between humans and the mouse is actually identical, and therefore we really believed that introducing the same mutation would really give us the same phenotype. And subsequently we used just a standard knock-in strategy which included the mutation in exon 11 and then selectable markers which were flanked by lost key sites, achieved homologous recombination, and then subsequently we had to remove the near cassette because the near cassette in vivo generated a hypomorphic phenotype. And so this is actually something that we have seen throughout the KIT gene. We have made many knock-in mutations, and but anyway, so then what did we get? We actually reproduced the human disease in all mice. Penetrance is 100%. And these are tumors that are probably about 2 or 3 months old. This is a tumor lesion in the cecum or the cecal junction, iliocecal junction, and these are just different magnifications. There is a low mitotic index, as is seen in this type of tumor. In addition, we have patchy hyperplasia in the myenteric plexus region throughout the entire gut, that is from the lower esophagus all the way to the colon. And so here is a normal section, a cross-section through a normal gut wall, stomach wall, and the myenteric plexus region is here. You barely can see it, whereas over here, this region is really quite hyperplastic, and you still can see the muscle layers, the cervical muscle layer and the longitudinal muscle layer. It's the same in the colon. Now, histologically, the GIST lesions we find in mice and are identical with those, indistinguishable with those found in human familial GIST. And here is a human familial GIST case we had at Memorial, where in the vicinity of the lesions there is also hyperplasia, and the morphology is really identical. So then we have produced a mouse model for the study of constitutive activation of KIT in oncogenesis by a knock-in strategy introducing an exon 11 activation mutation into the mouse KIT gene. Histological findings indicate patchy hyperplasia of KIT-positive cells in the myenteric plexus region throughout the entire gastrointestinal tract. Neoplastic lesions indistinguishable from human GIST are observed in the cecum. and sometimes in a large intestine. This is a little different than in humans where the tumor lesions are found in the stomach as well as in the small intestine. But we have to remember that in the mouse, the anatomy of the cecum is actually very different. It's very dark. So it could very well have something to do with that. And at this point, we don't really know the answer. Now, what are we doing now is we are interested in studying the mechanism of oncogenic KIT signaling. In this tumor, use therapeutic intervention, targeted inhibition of KIT and or critical downstream signaling molecules, and we are developing mouse models for imatinib resistance GIST. Now, the tyrosine kinase inhibitor, Gleevec or imatinib, everybody presumably knows about, is a specific inhibitor of the allele tyrosine kinase and KIT and the PDGF receptor. It is used to treat in the clinic chronic myelogenous leukemia very successfully, and it inhibits the an inactive conformation of both kinases. Now, when we treat animals with Gleevec, what we see, this is a time course analyzing cell proliferation index and increase of apoptosis. There is clearly clinical benefit. Now, we assume there is a lot known about KIT receptor signaling in vitro systems by us and by others, a lot of other people. It is known that KIT activates several distinct signaling cascades. SARC family kinase signaling, PI3 kinase bindings, PI3 kinase signaling, the activation of the RAS MAPcase pathway, and STAT transcription factor signaling. Now, if we treat the animals with Gleevec, then we may block the activation of these components, and thus we could actually get insight about the mechanism of KIT signaling in the tumor. Now, what we first see is that upon treatment with Gleevec, that autophosphorylation of the KIT receptor is downregulated. On the left side is placebo, and on the right side is imatinib treatment. We use a short-term treatment, 6 hours. The pharmacokinetic is really quite fast, and so the biochemical analysis we do is always being done at that time. Whereas for the clinical evaluation, we wait the 7 days to 2 weeks. Now, here we have analyzed several downstream effectors of KIT signaling in a PI3 kinase pathway. Serine ACT phosphorylation is downregulated, mTOR, and then quite prominently ribosomal protein S6 phosphorylation is inhibited, and then in addition STAT transcription factor phosphorylation is diminished, as well as MAP kinase signaling is also diminished significantly. So in GIST patients with GIST, imatinib therapy achieves a partial response or stable disease in 80% of the patients. However, often patients develop resistance to imatinib therapy. What is the mechanism of the resistance to imatinib? And then the identification and validation of new therapeutic targets, of course, is quite important. Christina Tedesco at Memorial characterized several imatinib-resistant GIST cases and found the acquisition of second site KIT mutations on one hand in the internal part of the kinase, and one of these mutations is a so-called gatekeeper mutation, which had been found in the BCR-ABL case as well. Second, there are activating mutations in the activation loop, which presumably stabilize the activation loop, and imatinib is not able to inhibit the activated kinase. Now, the strategies for the treatment of imatinib-resistant GIST. First, one may directly inhibit the imatinib-resistant mutant KIT receptor. Alternatively, critical downstream effectors of KIT signaling could be targeted by appropriate inhibitors, or an inhibitor may actually do both things. And an example of the last is the zatinib, which is an inhibitor that was developed as a SARC family kinase inhibitor, as well as an inhibitor. It also inhibits FAK, and has activity against imatinib-resistant BCR-ABL. It has also activity against some imatinib-resistant KIT mutants, including activation loop mutations, and thus is conformation tolerant. Now, in this context, we first wanted to analyze the effect of imatinib treatment actually on SARC family kinase signaling. And that is where we made a surprising discovery, whereas imatinib, as I showed earlier, downregulates KIT autophosphorylation, but SARC family kinases are activated. In addition, FAK is activated as well, and this seems to be follow a dose response in terms of downregulation of the KIT receptor and upregulation of SARC family kinase activation. Now, the SARC family kinases that are contained in that complex actually include SARC, LIN, and FIN. Now, these kinases are not found in an immune complex with KIT, and therefore we really didn't know what was happening here. However, we had fortunately included an analysis of FAK, which also showed that FAK was activated, and FAK has a critical role in integrin signaling, and is actually a major player, a critical player in integrin signaling. It interacts with the integrin beta chain, and upon integrin clustering, autophosphorylation phosphorates tyrosine residue 397. This serves as a docking site for SARC, which in turn activates the main tyrosine phosphorylation sites and fully activates the kinase. So then, analyzing all the different sites, we find that in imatinib treatment, upon imatinib treatment, everything is activated. So it seems that in the GIST mouse model, imatinib treatment induces a compensatory integrin signaling. And the question then was, what does the zatinib do, which is an inhibitor of SARC family kinases as well as of FAK? And just to go through this last slide, I would like to show, namely that the zatinib treatment of animals still leaves tyrosine 397 phosphorylated, but everything else is is inhibited. And this indicates that actually the zatinib would be a good drug to really be used in the treatment of this. However, when tumors or animals are treated with zatinib, early proliferation is being inhibited, apoptosis is not being induced as in this. Now, we therefore, oops, what's happening here? Combination treatment actually increases the response in our mouse model, and the reason for that is that imatinib, in addition to everything else, inhibits STAT family inhibition, whereas the zatinib doesn't do that. So this points us, in addition to imatinib upregulation, to that STAT3 activation may actually have a role. So I think at this point I would like to stop and thank the people who did the work. Ferdinando Rossi and Jasmin Jostat did most of the biochemical analysis that I showed. Benedict Bosbach has made a number of new histolines, actually imatinib-resistant alleles, and Shayu Despande and Huichen are other members in the lab. Thank you.
I'd like to thank all the speakers.